Okay, well, I guess to start, welcome everyone to the second tournament talk. This tournament talk will focus on the Atlantic Anglers Challenge. My name is Sean Simmons. I'm publisher and founder of the Anglers Atlas and Mycatch. And today we'll be going over some of the results from the 2019 tournament, as well as having Trevor Avery and Leah Creaser present some of the results and findings from the event. But before we get in, what I'm going to do is, and let's make sure I'm not sure I know this technology well, so we'll test this. We're going to do some polls tonight just to get a sense of who's out there. And the first poll is to which group do you most represent? And so I'll put that poll up. And hopefully you're able to answer that. And while you're answering that, I'll tell you a little bit more about who we are. So I founded Anglers Atlas about 20 years ago, over 20 years ago. My background's limnology, which is the study of lakes. And we cater to the sport fishing market by providing free fishing maps to anglers across the country. Now, about 3 years ago, I was down in Victoria at the World Recreational Fisheries Conference and presenting some of the results of all the data that we've been collecting. And had a good chance to talk to a number of researchers. And one of the challenges they expressed, a major challenge, is it's very expensive to get good data on the state of our fisheries. And so what ends up happening is only a handful of places get sampled a year, and most of our fisheries go unmonitored. So it really inspired me to think, well, we've got about 1 million anglers who visit our platform each year. So why don't we see if we can have anglers actually help solve and address some of these problems? So we launched the Mycatch app in 2018 as a way of just personal catch log for anglers so they can record their catches. And we share this data anonymously. And I should emphasize, secret spots stay secret. We don't publish the location, latitude, and longitude of information. But we do anonymously share the data with researchers who sign data sharing agreements to also not release those detailed locations, but allows us to get a much better sense of the state of our fisheries. And then COVID hit last year, and we work with a number of tournament partners, both Dallas Kirkpatrick, who's our director of sales, and Jim Clark is our tournament coordinator of Calgary. Realized that all the tournaments across the country were being canceled. And so we thought maybe we can adapt our Mycatch app to actually run COVID-safe catch photo release tournaments. And that's when Trevor introduced me to Jeff Wilson with the Striper Cup. And we kicked off our very first tournament called the Atlantic Anglers Challenge. And so that's basically how we got to this point. Now, the purpose of these talks is really to get a better sense of all this data we're collecting. We want to be able to share it back with the anglers so we can not only show you the research that's being conducted, but really learn from anglers and get a sense of what's important from an angler's perspective on what they're looking for, how we might be able to help better cater to the angling needs, and in the process, improve our ability to collect data for research and science purposes. So that kind of gives you, in a nutshell, what our purpose is here. And without further ado, what I'll do is I'll just highlight the agenda for today, basically. So there's my brief introduction. I'll end quickly. And then I'll pass it off to Jeff. And Jeff will talk to you a little bit about sort of his experience, certainly with the Striper Cup and what we did with the Atlantic Anglers Challenge, and talk a little bit about things from an angler's perspective. And then we'll feature two fisheries researchers, Dr. Trevor Avery and Leah Creaser from Acadia University, who have been working on striped bass research for a number of years now, and they'll be able to present some of their results and some of the unique findings we've learned from this event from last summer. And finally, we'll finish things off with a question and answer session. And we really want to learn a lot from the anglers. We think that this is important to have a two-way discussion. I think anglers have as much to teach scientists as scientists have to teach anglers. So it's really important to have two-way dialogue. And what we'll be doing is emphasizing them. If you have any questions or comments, please enter them into the, I think there's a questions section, Q&A section that you can add in a question there. And please, at any time, just add in those questions and we'll be addressing them as we're working through this. I think that's all. So maybe what I'll do is I'll just pass it off to, the poll. So I think what I'll do is I'll end the poll. And let's see, I think I can share results. Share results with the poll. So it looks like anglers, the majority are 67%, researchers at 24%, managers at 5%, and other at 5%. Excellent. So I'm thinking, I don't know how this works, but let me just quickly screenshot that. I think I can stop sharing, and then we'll jump into poll number 2. So while Jeff's talking, if you can, poll number 2, launch poll. Yeah, so poll number 2 is to really get a sense of what areas of fisheries science you're most interested in. And so we'll launch that poll. And without further ado, I'll pass it on to Jeff. Jeff.
Hey, well, good evening. Thank you, Sean. First of all, I'd like to just give a little background on how this Atlantic Anglers Challenge started. Like Sean mentioned, that he and Trevor were at a conference, and then somehow they figured they should get introduced to myself, and I was the gasoline on the fire to get this thing going, I think. And so really the Atlantic Anglers Challenge was an Atlantic-wide fishing competition designed to jointly support COVID relief and recreational angling research through engaging anglers and citizen science. So the thought was that if we could get people to put all their catches in, and certainly after working with Leah and Trevor and that at the Striper Cup, which is the largest fishing event in Canada, certainly saw how science should be react with tournaments. And so that gave us this idea, and I figured it was a great cause. And we found out basically the major challenge by most biologists is that to try and do field work in multiple bodies of water across everywhere in a given province or area was almost impossible. So the only way to really get that scientific data was to engage the anglers, to give it to us as research scientists, and to be able to take that data and play it back to them. And that was the major front for this. So we thought the best way to do that was to maybe get some great brand manufacturers to give us some small enticements, and a big reach out to some of those: Dorian Outfitters in St. John, Shimano, Rods and Reels, Garmin Fish Finders, makes it easy to find them. You can catch them. The Real Deal down in Nova Scotia gave away a charter. Surrett Battery rolls batteries out of Nova Scotia. I personally hand delivered all those batteries to the people around Atlantic Canada. And of course, the Striper Cup was an event partner. And we certainly look forward to this spring to be able to use this app during the Striper Cup. That was originally our plan. And when the Striper Cup was canceled, we felt that it was a great opportunity here to get these recreational anglers doing some social distancing. So we thought that was a great thing. And so we went ahead, and Leah was kind enough to loan hundreds of hours of her time behind the scenes, because her Professor Avery there was telling her to get going. I need you to do this. So we got that. So tonight we have Emma Boyce here from the St. John Regional Hospital Foundation, and she's the community engagement liaison. Is that right, Emma? I think it's
coordinator, but it really doesn't matter.
Coordinator, okay. And so we were really lucky that our plan was to give half the money and half the prizes. Thankfully, Sean's such a nice guy. He decided just to make it an even amount of money. So we wanted to get Emma on tonight, and we got a nice check presentation that we've already done. But I did this for tonight, Emma. Look. So we got $1,000.
That's very exciting. That's definitely more exciting than an e-transfer.
Okay, so look, I'm gonna pass it down to you.
Thank you very much.
Yeah. So that was our donation to. Maybe Emma, you could tell us a little bit about your foundation and how that works.
Yeah, for sure. Well, first of all, we were just so excited when you guys reached out and chose us to be the recipient of the funds. I also was extra excited because I am an Acadia alumni as well. So that was a little extra special too. And, I'm always so inspired and grateful to see our community come together to support their province's healthcare, especially in times like these. And the funds that you guys have raised and donated will help support a myriad of advancements in our province, such as capital equipment and technology, research, education, patient comfort, and mental health. So from all of us at the foundation, we would like to say thank you for your generosity and especially your creativity that came with hosting an event during a pandemic. It's not something that is done easily. So thank you very much. And it is extremely helpful to everyone in the province. So thank you
Well,
again.
and that's Emma. So you basically, this money goes all through the province, eh? That's basically how it works. Yeah. So I know,
Yeah,
although it says
so
the St. John Foundation, but it's really distributed province wide.
well, so it goes to the St. John Regional Hospital Foundation, which then goes to the St. John Regional Hospital. But our hospital is seen by patients province wide and in Atlantic Canada. So, like, I have a couple facts. Like, our Heart Center serves nearly 1 million people all throughout New Brunswick and PEI. So, people from all over New Brunswick and PEI come to our Heart Center. Same as our oncology program, same as our Clinic One. People from all over New Brunswick come to our hospital in St. John because we do have some of the top healthcare and some programs that just some of the other communities don't have.
All I can say was really lucky you answered the phone that day, or we were moving on to the next hospital.
Well, I'm extremely lucky that I heard it ring then. So
yeah, well, I think what we needed to do was just to find. We wanted to do this as Trevor and Sean and I. We figured that would be a good method to get it going. So certainly would like to thank you, Emma, for coming on tonight for sure, and congratulations. We're hoping we're going to run this again next spring, and you're going to post a fine picture of me handing that that $1,000 check across there. So big thanks to Sean Simmons and Dr. Avery and the Striped Bass Research Group. They did an amazing job this summer to certainly work along that.
Yes, thank you.
One of the other things, when you've seen our challenge, you can see that lovely striped bass there. So just so Emma, that's what you've been looking at all week. So maybe next year during the Striper Cup, you can come up to Miramichi and just have a look and see what we really are all about.
Yeah, that would be awesome. I have actually grew up fishing. I'm not very good, but I would love that. It would be awesome. And
yeah, I don't
really
like fishing. I only go catching. That's my rule. Catching is what I do. Fishing is boring.
Okay.
Yeah, so we'll get you out. Well, thank you so much again, Emma. We certainly appreciate that, and we look forward to maybe a repeat of this next summer as we figure out how we can get this going.
We would love that. Thank you very much.
Yeah, no problem. Everyone. Thank you so much.
Bye.
So just to wrap up from our Atlantic Anglers Challenge talk, we had great participation. Leah's going to go back and put a lot of information out there to the anglers. Any of the anglers that have any questions will be willing to give those answers. I got some real good questions I'm waiting on, so certainly going to have that. And I think, how's my time, Sean?
I think it's pretty good. I think we're spot on.
You gave me 15 minutes, right?
Yeah. Well, I think you actually got a couple more minutes if you want to keep going.
So, I mean, one of the things that I think from an angler's perspective was the Atlantic Challenge kind of built as the summer went on. And, we had groups like Garmin and Shimano and Rolls and prizes. And I think what we did last year, we bit off a bit bigger chunk than we could really deal with. But I think we're going to drill that down. But it's funny, I talked to some of these folks already, and I actually got some grief from some other brands that said, hey, how come I wasn't involved in this last year? And I had to send them the email where they turned me down. So, I said, so we're getting this is good. I said, so hopefully we can get some engagement. I think it's really important. And we're trying to get some pilot projects off the ground with different provinces and those sorts of things with Sean's help. And certainly the app came such a long way in the summer from how we started that. So, I mean, my hat's off to Sean and his development team. One of the things that really boggled my mind, I think, is every Monday morning we were meeting for the development team, and Sean runs a pretty tight ship out there in BC. So there's just the work behind the scenes. I think it's really important for people to understand. And that's why we called it a challenge, because it wasn't just a challenge for angler to catch fish, but for us to get the app working properly and everything working together. And I hope it started a lot of stuff that will certainly carry the app further on down the panel with this. And so certainly my thanks to you, Sean, for allowing us to use that. And we certainly are looking to see the Striper Cup use that as our shore division or our individual division this May. So our plan is to use the app to not only get the creel data that we need, because DFO doesn't know how many people fish bass in Atlantic Canada, so it'd be kind of nice to know, but also for us to have that continuous leaderboard every day in the Striper Cup to try and see how everybody's doing. I talked to one of the sponsors today from Jeff Morris from Morris Wholesale. He was telling me how his kids just love it, and he couldn't believe that we might be using something with a live leaderboard. So I can really see the kids getting interactive with that sort of thing. So really look forward to that kind of thing. So anyhow, so I'm down. I actually have 2 minutes left, Sean. So I'm gonna give Leah a little bit more time tonight. Everybody, they can listen to me all the time. It's really important to get the science out there. So I'm going to turn it back over to our host, Sean.
Yeah, well, we've actually got a question for you. It says, Jeff, has DFO or DNR expressed interest in getting involved in supporting this kind of program to help aid in species management on our waterways? From Donald Patterson.
You know, that's a great question. We have engaged the province of New Brunswick, as Sean knows, to see if they are interested. They certainly, the report from science was that they really liked it. So we are into some discussions with them as well. I think, Sean, you've been speaking to DFO as well about a pilot project. So governments move slowly, but one thing we did learn was there was 0.04% of anglers filled out creel surveys in the province of New Brunswick. So the Atlantic Anglers Challenge actually has more data than the last 10 years combined of the Department of Natural Resources in the province of New Brunswick. So our data is pretty solid. So hopefully, yes, Donald, I think the province sees that there's some value there, and I think we should see something in the near future.
Awesome. Thanks for that, Jeff. And just to end the poll, and let me, I guess, share the results. So in terms of what area of fisheries science interests you the most, management and conservation are in the top assigned position at 40% each, followed by research and then biology. So what I'll do is I will now go to the third poll, and this is a poll really focusing on from a biology perspective. What are the things that interest you most? And I'll launch that poll so people can fill that out while we jump into the science part of this question. So I'd like to introduce Dr. Trevor Avery and Leah Creaser from Acadia University. Take it away, Trevor.
All right, great. Hi, everyone. I'm going to share my screen here if I can, which I think I can. There. Hopefully everybody can see my screen. No, that didn't work. I didn't hit the actual share button. Now you can see my screen. Just give me a okay if you can see my screen, because I have no idea whether you can or not. Okay.
You're good.
Good.
Yeah.
All right, we're good. So I was asked to provide some information on general science and a few pictures and things like that. And I'll touch upon a bunch of those themes that were brought up there, fish biology and conservation, and how we do some of this work from a technical perspective and a research perspective. And a lot of this is couched in citizen science, and that'll lead into what Leah is talking about. I'm going to move sort of quickly through these slides because I've got a few slides here. So I have this group called the Striped Bass Research Team, which we formed back in about 2008 or so, just around the time that the Striped Bass Research Association was getting on the go. So most of this talk will focus on striped bass, but we do a lot of other fisheries monitoring work around the province as well. So if you don't know what a striped bass is, it's these nice big fish here out in the Atlantic coast. Sean's going to come out one of these days and fish these. Jeff's caught quite a few, and I see a few names on the list here of people that I know have caught some of these from what went on in the Atlantic Anglers Challenge this year. So these grow to be a pretty large size, and there was a conservation piece on the survey there. Right now, I'm participating with a few others in actually looking at this whole idea of where the striped bass are around the Maritimes, and putting that picture back together. It's called a COSEWIC report, and it's quite a big deal. It'll take probably another year or so for it to come out, but at the end of the day, this is one of the big reports that's used for some of the management. And again, it could change some of things like, or at least inform management and conservation rules for fishing, et cetera. So that's on the go. So we do a lot of work with citizen science and towards this idea of conservation. And so here's just a couple of pictures of some of the things that we do. We go out to a lot of beaches and talk with a lot of anglers, do things particularly, and Leah will talk about one of these that we did last summer during COVID, going out and interacting and showing people sort of what our science is all about. And so we can kind of sum this up and talk about it as conservation, monitoring, and stewardship. So the stewardship part is essentially all of the anglers out there doing their sort of work and being stewards of that resource. And so we rely on that a lot to gather information for monitoring purposes that we then can use for conservation purposes. So we sort of created this community over the years, and it's taken quite a while to sort of get up and going, but we have a lot of people now on our Facebook page, and a lot of interactions when we post about the things that we do. Mainly we're interested in doing tracking of fish, but there's so many offshoot projects that we can also work on. So I'm going to talk about a few of those today, and a few of these people are going to come up in the talks over and over. So one of the big things, and Sean mentioned to me that in some of the other tournament talks that they've done, the real thing that people are interested in is sort of where fish move. So I'll sort of focus a bit more on that. There's two main technologies to looking at movement. And the first one is to put these sort of low-cost dart tags in a fish. There's a little striped bass that was dart tagged. This is probably Danielle Quinn's hands here from a number of years ago. And the second technology is to use an acoustic tag. So this is a little tag that you can put inside of a fish, and it's got a battery in it, and it basically gets picked up by a receiver that are hung off of buoys or put into structures around out in the ocean. So essentially, the fish get picked up on these, and then you can tell where they move. So there's advantages and disadvantages to both of these. These acoustic tags cost about $400 to $500 a piece, so they're quite expensive, whereas the little plastic tags cost about $3 a piece. So we use these to look at where fish migrate and move over long distances, and a lot of the time that's to search for food. Lots of early studies out there on dart tags and T-bar tags, so there's sort of two subtypes of these tags. We use mainly dart tags. T-bar tags don't stay in as long. And more recently, things have moved to this acoustic picture, but it's super expensive. And then on top of that, we take fin clips, and we use that for genetic analysis to give us some sense of where these fish have come from. So even though you might have caught a fish, you're not quite sure whether it's from a local population or maybe it's migrated in from somewhere else, or maybe it's just on a trip for that day. And we found all those sorts of things in our analyses. So our objectives for a lot of the stuff we do are to just essentially collect striped bass samples. And these come in various different ways. We have a carcass program. We have people that have scale envelopes and they collect scales for us. We have other people that send us pictures. Our Facebook page often has pictures throughout the year of people catching tagged striped bass. And then we also work with this guy, Derek Brothers, who has one of the larger Facebook pages for striped bass angling. And we work a bit on the data that people post to that as well. And so all that stuff we've been working on over the years. So I'm going to talk a little bit just of a couple of slides on each of these topics here, just to give you an idea of sort of the breadth of stuff that we do and the way that your help with this information can help us. So we go to a lot of derbies. This is a picture from Jeff's big derby there, the Miramichi Striper Cup. I can't remember what year this is, but these are the fish coming down off the weigh table, and then we grab the fish, and you can see that there's 5 people here all doing, there might even be a 6th hidden in the back there, but 5 people all doing something different on this fish. And so within about 10 or 20 seconds, we can have everything processed that we need for this fish, scale samples taken, length. You know, there's double duty here, writing something down and taking a scale and putting it in an envelope for the next fish. I mean, it's just efficiency is at the top notch here with this crew. We do a lot of hands-on research and outreach programs. So we go out to the communities and we show people sort of what we're doing. So this is from many years ago. Peyton here was age 6 at this picture. This is, geez, this might be going back 10 years now. And we went out and did some scale aging and show people how we age scales and where we take scales from, et cetera, on the fish. And here's an example of Julia Whidden out taking some scales from many years ago. Actually, it might not even be Julia. It could even be another researcher I had back, way back. This is us going out to show people how we do the tagging. So this is sort of a tagging demonstration and also give people a hands-on go at it. And this is leading up to one of our newest programs called the Tagging Ambassador Program, where we go out and train people to actually do this tagging part. We've got a list of maybe 6 or 8 people for next year. We were hoping to get things off the ground this year, but of course we couldn't meet with anybody because of COVID. So we've done a bunch of tagging workshops. Some of these were done through Mi'kmaq Conservation Group and First Nations communities, Nova Scotia, New Brunswick. We've had over 150 people participate in those early workshops, and now we're sort of honing that down to where we're actually getting to the training. And I noticed a couple names on here. Owen Marr is one. He's one of our tagging ambassadors. He's all trained up and puts tags in for us and probably puts in 100 or so tags a year. So there's a little bit to it. We have to go through some lab work. We have to go through some protocols, et cetera. And then we actually do hands-on training where we take people out to show them tagging. Another aspect that we do is catch logs, and this is a sort of a more simple thing that people could do, and you can do this 2 different ways. The first way is we can provide you with a log book, a waterproof log book where you can record your catches in, and then we get that data at the end of the year. And this is preferred by some people. And the Atlantic Anglers Challenge was actually a proof of concept to see whether we could take something like the Mycatch angling app and use that instead of the log books. And we're pretty much to the point where I think we can do that. The only thing we don't get is with the catch logs, we often get people provide us with scale samples as well. And so there is a little bit that we still have to work out on that front. But essentially, this is a way for you to participate, to give information just as you're out there fishing. And all of this information gets compiled and used in various ways. So this is us out doing some fishing at the Shubenacadie River Derby, and this is up at the Miramichi Striper Cup. And Leah's going to talk a little bit more about our COVID days here. This is our Atlantic Anglers Challenge that we did this past summer. We take those catch records and we look at sort of where fish move. And so we have lots of high catch records in all of these areas. And then we have areas where we anticipate we should be getting catch records, but we don't get too many catch records. Now, there's a little bit of a divide here because in this southern Gulf region, this is sort of all worked up by DFO, and all of these catch records we have, even up into the Petitcodiac River here, are all tags of fish that we tagged. So we tagged fish mainly in this area, and we've caught them down in Annapolis Royal, for example, and one of them got caught way out here in Bredor Lake. So we know these fish move, that's for sure. So how many fish have we tagged? Well, we've tagged, this is the bass that we've sort of handled over the years up to 2017. We've got a few more years here, but this was a specifically directed program over these years. And so we worked in a commercial fishing where got to handle a lot of fish. We tagged quite a few fish over those years, and these are the number of recaptures that we've gotten from those fish. And essentially, the recaptures part is where the citizen science anglers come in, the recreational anglers. And the numbers bounce around a lot, and part of that is due to the uptake. We have to really get out there and do our outreach and our awareness things and post a lot to Facebook and get people enthusiastic about sending in these recaps. We know there's people out there with a tag in a tackle box that hasn't reported it. And those are the sorts of things that we really want to emphasize, that this is really important to us, and it's probably really important to the conservation of the species to sort of return those recaptures. So this is just a little video here of Derek Brothers doing a little bit of tagging training with Jackson Yang. I don't know if this is going to play. I might play a little bit of it just to see.
A little light. I'm saying, Trevor, that's where the Atlantic English Challenge and the Atlas would really help, eh? Taking those pictures and getting that done really quickly. And
you get the idea there anyway.
Yeah. Did Jackson get good marks for that?
Yeah, so he goes out and does a fair bit of our training right now, and it takes a little while, and we do have a permit in case fish get hurt or whatever. So, I mean, we know that there's going to be a few fish that are probably going to be in rough shape from the training part of this, but once we get past that, then we're going to make much more gains. And so there is always a little give and take with conservation work. So we've got quite a few fish tagged. These are sort of the areas places that we get lots of returns from. So lots of different places, which is great. And we can use these tags to give us all sorts of information. And a couple of the really interesting ones right now are looking at movement patterns and estimating growth rates of fish. And this is something that Leah O'Halloran, who's actually on the group here watching this now, is just finishing up as part of her thesis work. We also take a little fin clip or a couple of scales, and we can do genetic analysis with it. So this is some information that we've done 2 papers now on genetic analysis, and we've got a third bit of information coming. So this is sort of up the whole eastern seaboard. And what we're seeing here are little pie diagrams, and the different colors mean the different populations. And so the pink population here is the Shuby River population, and so most of the stuff from up in Minas Basin is Shuby. The brown ones here are ones that we figure are from the St. John River population, so this is a genetic group from there. And then the blue ones and the green ones are from the Chesapeake and the Hudson areas. And so these are U.S. fish. And so we see a few U.S. fish that you get up in the St. John area and very few that we see coming into Minas Basin. And so we listen to recreational anglers. They ask questions, you know, oh, I think it's all U.S. fish that come up here in August and all those sorts of things that we've all heard and talked about. And so we actually go out and try and answer these questions. Is that the case? Here's other work by Leah O'Halloran, who's done some work in Minas Basin. And so these are all fish from the Minas Basin, these green ones. And we see the green ones are all Shubenacadie River fish. So this is sort of our known good samples. These are our samples that we didn't know where the fish was from. We have 1 stray fish out of 130 samples or so that came from Chesapeake or Hudson River. So 1 U.S. fish out of about 130 or so. So, you know, less than 1%, fewer than 1% of the fish that come up in the Minas Basin over the summer are likely U.S. fish. And then we have a little bit of a sample here of Labrador fish. There was an event in 2017 where we got some fish that moved up to Labrador. We got some samples, and it turns out they were all from the Miramichi River. So really interesting stuff that we can find out through genetics. There was also a tagged fish caught from the Miramichi River in Labrador. So we knew right away those fish are from Labrador or from Miramichi. We work on fish stress. This is something we're looking at with Dr. Russell Easy at Acadia. This is some of the early work we've done with Hilary Dort, who's gone on to become a dentist. You know, no more fishing. And this was a sampling fish with looking at mucus. So essentially we can tell a lot from the mucus in a fish, and we can tell whether it's stressed. And so we've been working on that. It's a costly thing. It's $150 a sample. So it takes some time to sort of work through that. And the last couple of things I'll talk about are our diet analysis and contaminants that we get from that. And this, so we call this our carcass program. You've probably seen us promoting this. Hey, if you catch a fish, we'll come and pick it up. We like fresh fish. We don't care if there's fillets on them or not. Better if we have fillets for certain things, but it doesn't matter for lots of things. And what we do with this is we take out the stomachs for diet analysis. We take out the liver for essentially diet analysis, but done a very different way. There's obviously nothing inside the liver of a known thing that we can look at, something that it's eating, but we can tell by doing a thing called stable isotopes analysis, sort of what it's been feeding on. We take muscles for a similar thing, gonads for development, otoliths for aging, and growth, and scales for aging. And up here in the upper right is a picture, is a picture of an otolith of striped bass that we've dissected out. This is from a very small striped bass, actually a young of the year, one that's only about 10 centimeters long, and this is magnified. So this is a little inner ear bone that gets layers upon layers added to it like tree rings, and we get them out of the big fish, and we can weigh them and get an idea of how old that fish is. This is the male gonads, female gonads. These are some samples, and this is just part of our program. We probably have processed 350 to 400 fish over the last couple of years. And what we do with this is look at sort of the development of the gonad leading up to spawning time. We had a question from a recreational angler. I caught a fish in November and it had giant gonads in it. And so it was a female, had big gonads like this in it, and it was caught in November. And we thought, well, that's odd. Did they spawn at a different time? Are they spawning in November, or are they just gaining resources throughout the year and then sort of holding on to those resources throughout the winter? And that's essentially what we're finding right now. This is a little bit of our diet analysis done very differently. This is, instead of looking inside the stomach and saying, hey, that looks like a capelin, or that looks like a piece of lobster or something, we do this genetically. So we actually sample the genetic goo, we call it, on the inside of the stomach, and then we see whether there's pieces of DNA from different animals. And so all of these fish, there's maybe about 10 or 15 fish here. This is the proportion of things in its diet. There's obviously a lot unknown still. All the pink is unknown. So they're feeding on things that we don't know what they are yet. But we've only tested these fish. These are from Labrador. And we've only tested these fish for Arctic char, Atlantic cod, Atlantic salmon, and capelin. And we noticed that, yes, they do feed on different things and probably at different times of the year. This is the GSI, or Gonadosomatic Index. So this is the ratio of the gonad weight to the body weight. And that's how we can tell how much energy they're putting into the gonads. As the gonads get bigger, it's a higher proportion of the weight of the entire fish. This is around the spawning time. So this is what you'll see with spawning fish, is these high GSI numbers. After spawning, or leading up to spawning, there's a high GSI, and then it starts dropping around June and July because we're catching fish that are probably spawned out a bit. It levels off for most of the summer. Nobody catches fish in August, it seems. For striped bass, it's a little warm, so it's hard to get samples. And then leading up to November, we start seeing this increase again, so energy being put back into the gonads. So that picture is starting to become quite a bit clearer now, and that's all information from people donating carcasses to us, or mostly anyway. This is what we do with the gonad samples. We put them in a solution, put them in wax blocks, cut them into little sections, and then we can actually slice them up and look at them in a microscope and look at what the gonads actually look like. So what we're seeing here are different magnifications of a slice through a female gonad, and these are actually the developing eggs here. This is all these sort of large things with the nucleus in the middle. So this is the genetic material, and all these little white spots here, these white round areas, that's all the developing yolk that's being coalesced together to create that egg. And the other pictures here are early stages of the same thing at different magnifications. So we're really getting a good picture of what's going on with the gonads. Oh, I got some stages on here, not a big deal. And that's basically it. So this is the crew from a couple of years ago, went out and with Derek Brothers and a couple of others, and they all came home with a keeper that year. So thanks to all of our partners here that we've worked with over the years.
Trevor, I think you've got a few questions there popped up for you.
Okay, let me just stop sharing this, if I can do that. Stop share, because I don't get the questions. Yep. Okay, here we go. Let's see. Oh, where do I have to go here? Does somebody want to help me with this, DT?
Yeah, sure. I have the list of questions kind of written down, and
perfect.
Can give those to you. So we'll start with the ones with in the Q&A box. So Donald is asking, the school size in the Miramichi seems to have fluctuated in recent years. They believe the current number is around 350,000. And is that, do you think, a healthy, sustainable population based on current angling patterns?
Yeah, it's a really good question and a complicated one at the same time. It's really hard to estimate those population sizes. I think I deleted the slide that I had showing those that relationship that you're talking about. The values, that 350,000 is plus or minus, and we call that uncertainty. We're uncertain. It could be much higher, it could be much lower. And that's common in science because we can't census everything. We can't count every fish. And so we do this through an estimation procedure. So those values, the best we can do are those sort of ranges of values that we get. Is that the right number? Well, there's a report going to come out in a couple of months' time that I was part of with Department of Fisheries and Oceans up in the Maritimes region, or sorry, the Gulf region, where they've looked at modeling all of that population. And the idea is to come up with what's called a reference point. What is the minimum population from these estimates that we get that this population needs to be at in order for it to be sustainable? I don't know what the final number is. I know that it's definitely something that's been thought of at high levels. This goes right up to the minister. And it is something that's coming down the pipe, and it's probably going to change some of the rules. I don't know in what ways. So that's something that's not necessarily something that science does. We provide advice on those things, and then there's a management group that's going to that sort of weighs other things in there. So I think there's a long road left to go. My personal opinion, I think there's a lot of fish up there, but I don't know what a lot is historically, right? And so it's difficult to say.
All right. So there are a bunch of questions that have come up in the chat as well. So what we'll do is I'll give you the other questions from the Q&A box, and then we'll move on to Leah's part of the presentation, and then we'll come back to questions at the end just to make sure that we have enough time to cover
Yeah,
everything.
she may answer some of the questions in her presentation too. So
Yeah. Okay, so another question from the question and answer box. Nick is wondering, he caught quite a few fish this summer with a white scale-like substance on it. Do you know if that's normal or what might cause that?
it's probably a thing called ick, which is it's not a parasite. It's just like a fungus, and it's normal in fish. They just get it from time to time. It's something that's out in the waterways pretty much everywhere, and you'll just see at certain times when the temperatures go up and the fish get a bit more stressed, you might get a bit more of that in the fish. It'll go away naturally because the mucus that fish has a lot of antibacterial properties and antifungal properties in it. So it shouldn't affect the fish at all. If you get, if you're looking for nice fillets and you open up your fish and you have fillets that don't look good, like black spots in them, things like that, don't eat those. It's probably full of parasites of some sort.
All right, and the last question from the Q&A box. Mike says, I saw from your slide there are 5 different breeding populations, and I assume due to the striper cup involvement, that region is producing the most data for you. What population group do you need better data on? Basically, what population do we know the least about as of today?
That's a fantastic question, and I'm glad you asked it. The Annapolis River and the Petitcodiac River in that region are 2 areas where we just don't have a lot of information. We work with one fellow down in Annapolis, Royal now, who's why is Rob Weed. His name's escaping me a little bit, but anyway, he's down there and he does a really good job of promoting what we're doing and gives us lots of samples. But we're really missing sort of that catch data and that sort of monitoring aspect from that region. And the Petitcodiac River is another one. It's been opened up now for almost 10 years. And we're starting to see larger striped bass up in that area. So it'd be nice to know what's going on there. We're probably going to take our tagging road show up there as soon as COVID allows us and be doing some tagging fish with the Fort Folly Habitat Recovery Group. So, yeah.
Awesome. All right, I'll pass it back to Sean for now, and we'll come back to the other questions closer to the end.
Great.
All right, thanks, Trevor. That's excellent overview. A lot of information, like drinking from a fire hose. Really good, thank you. So what I'll do is I'll quickly end the poll and share the results with everyone. So basically, which fish biology topic interests you most? The top one, how many fish are there? Population dynamics. How does a fish's body work? It looks like that's not of much interest. Where do fish go and why? Tied with what conditions do fish live in? And how do fish act? So awesome results there. Let me jump into the next poll before I pass it off to Leah. And so this poll says, how would you most like to see this tournament data used? For fisheries research, for management, for conservation, or for your own angling interests? So poll is now live, and I'll pass it off to Leah.
All right, can you hear me?
Roger that.
All right, I'm gonna try this screen sharing thing, and we'll see if that works. Okay, I gotta somehow play it. Is it working?
It's good. You're doing good.
All right, cool. So yeah, basically I just put together a presentation as a whole overview of the challenge and kind of how we worked through it and any challenges we faced with the challenge, and then just some summary points at the end. So for anybody who's wondering. All right, so yeah, obviously as highlighted before, one of the main reasons we did it was because of COVID-19 relief. During COVID-19, we got the horrible news that, you know, we can't have derbies or challenges or tournaments in person anyway, and we have to social distance. So, and it also kind of left research at a halt as well, because we weren't even allowed in our lab for the longest time. Like, we were only working from home and working on our computers. We weren't even allowed to go in there to like get any belongings or anything like that. So yes, obviously we were able to part with Anglers Atlas and get this tournament going on Mycatch. So it just allowed anglers to participate for online fishing challenges in a COVID-19 friendly environment, and also give us data. Okay, so it's not moving. Okay, there we go. Mycatch, that is the app that was used. So it allowed anglers to sign up for the tournament and to submit their catches with the photo, species, and length information. One of the main points, as highlighted before too, is that the locations were confidential. So that was a big point that we found from talking with anglers, is a lot of people have their, their main fishing spots that they don't necessarily want to share that information with other people. So that was really good for them. And also the really cool part about Mycatch is when you're logging any trips or any fish that you're catching, it doesn't matter if you have like data on your phone or not, you could log them offline. And that's just a picture from the tutorial that Jeff did for a video on how to use your phone and fish safely at the same time. So the challenge was the first virtual fishing tournament in the Atlantic provinces, as far as we know. So it was obviously Atlantic province wide. It ran from May 2020 to October 2020. There's a $20 entry fee, and the money went to the St. John Foundation. So it basically included any species of fish. There was no age requirements. You didn't have to be a pro angler to enter. You could be just a kid, trying to win something. Locations are confidential. So there's also a whole bunch of challenges that included not only like the largest length or the longest length, it was diversity as well, different regions, seasonal, and just overall challenges as well. So for data collection, so obviously everybody downloaded the Mycatch app, and then they were registered for the tournament. And once they were registered and they paid their fee, they were able to start to participate. So once the participants started fishing, they were logging all these photos of their fish and logging their catches and their overall trips. And all that information was coming into us as scientific data. So basically, as those photos were coming in, we had to go through them one by one, and it was fun times. Nervous laugh about that. There's a lot of trial and error with that, but we got through it, and I'll talk about that as well. And basically, once we got through that, then the data was extracted and put into a program. Thank you, Danielle. And then we organized, formatted into Excel spreadsheets, and then we're ready to take a look at it. So for the approval process that I just nervously laughed about, so we ended up having to make a flow chart, obviously, so that it wasn't just me or another individual going through them. We kind of wanted to have a process that we could all go through and make sure that we're following the same things. So we made the flow chart, and even with our eyes seeing the flow chart, it was great. But then the anglers didn't necessarily know how we were approving or rejecting their photos, which could cause some conflict as well. So I just decided to make a poster and just put that up on our social media, and it was pretty good. So after doing that, because we've seen a lot of like trends in certain things, like not a clear photo, or you couldn't see the full length of the fish, or just little things like that. We couldn't say yes for sure that is the length of the fish, etc., etc. So we posted up. And after we did that, it was pretty good. Like all the anglers, we didn't see very many like issues with that after. So I guess the good thing about this is you could be entered for like your species specific prizes as well, like diversity as well, and length. So it's good because, even though those people uploaded those photos with the fish, we could identify that, yes, that's an eel, or yes, that's a striped bass, but we couldn't see the length. There's no point in just rejecting the whole fish. So it was great. We could do that way. So we definitely, through the challenge, encouraging and teaching our anglers. It was really fun. I found it really fun. So as I said before, there were some inconsistencies with uploading the fish correctly. And again, just pictures missing requirements and missing data when uploading. And so at first we were reaching out individually to these anglers, and we were just talking them through, and they were really great because, I mean, it's our first time doing this whole thing, and I'm sure maybe it was their first time on an app doing this. So it was really great to kind of make that connection with them and just correct it so that they were able to have the fish entered and not be rejected. So then again, my thinking is, why don't we give that information to them? So made a couple more posters just so that they could have a reference guide kind of to say, okay, this is all in my picture, we're good. And yeah, again, because of the posters, not trying to toot my own horn, we didn't really see much of that happening again. So this is just a couple rejections. I thought they were funny, so that's why I put them on. Obviously, you know, that's not a brook trout and that's not a northern pike. So those are cases where we'd have to reject all of it. And then on the other side, you know, there's a couple examples of, we know what species it is, but we can't necessarily guarantee that that's the length, and especially with the eel. All right, so for promotion, so again, COVID-19 kind of restricted every outreach aspect. So what we did was just like plugged our social media. So created Instagram and a Facebook was created, and everybody worked really hard together to stay on top of the social media. And that was it ended up kind of being like the main source of communicating with anglers, because I found that they weren't, I don't know, just email is a bit more like, I guess, more work, if I'm just going to put it that way, opposed to like Facebook Messenger or anything like that. So it was really great to have those platforms, and it responded really well. So some outreach. So yeah, Trevor stole my picture, but I have a picture on there. So yeah, once the restrictions started to ease up, like what a relief, we were able to do a couple outreach projects. So we basically blasted social media, and we were just like, we're gonna be here, like come on out and see us. So basically, like that top picture, we just went down to Five Islands, Economy, Parsbro area, and we just talked to anybody that would allow us to talk to them. And we found out where like the hot fishing spots were, and we set up a dissection table. So as people were coming back into the boat launch with their catches as the tide was going, we had our station set up, and then we had just a fillet station set up for them, so they could just fillet their fish and then give us the carcass, and it was great. It worked out really well. We got a lot of carcasses from that, and we were able to promote the challenge at the same time, and also just make that connection with recreational anglers. And also, like, they're like, what do you need all this for? You know what I mean? And as Trevor like had all those slides on everything we use a carcass for, we were able to do it right in front of them and talk to them while we were doing it, and like basically just show them everything we could do. And that kind of a lot of interest. Hey, well, what's this AAC? Like, let me sign up for that. If I can help out that way, that would be great. So it was really good. And so then we were also able to gain sponsors through harassing people. No, just through outreach, we were able to gain a couple sponsors, like Real Deal Charter, which is out of Parsbro. And so because of Real Deal, basically they were able, through talking to them, they were able to donate like a whole charter fishing trip as one of the big prizes. And also they're like a big family name down in that area of Nova Scotia. So they also had family that owned the Davis General Store and Fish Market, which was basically like the main bait shop for all of that area, like Economy plus Five Islands plus Parsbro. Like everybody went there in the morning to get their bait. So that was awesome. They were able to advertise our posters and everything for the challenge all on their bait shop and within the store in general. And they ended up giving us a whole bunch of prizes to give out, like a lot of they gave us rods, they gave us reels, they gave us line, they gave us like just endless things. So that was really good. And they were also able to take Josh and myself out on a couple charter trips, which was a crazy experience, but it was really fun. And the whole time we were there, we'd just like tag and fish like crazy because it was a whole different experience, I would say. Like we were there for 2 tides. We were out on the boat for 16 hours. It was crazy, like each time. And we were just bringing them in, tagging them, letting them go. So then like that next time we went, we were able to train Anna and Charlie as an ambassador to help out with the striped bass research team. So we made a whole bunch of connections that way, and I think that was like an amazing success. So citizen science, I know that's probably the main topic here. So overall, like goal was to have the challenge and develop that relationship of awareness and citizen science, and basically to have that rich information from anglers, you know, who don't necessarily like want to, tell you where their spots are, I guess. I mean, from their view, you know, you're just some random person being like, where do you get all your fish? So this way it was really good. They could do it confidentially and, you know, just feel okay about it while competing. So that was really good. And so yeah, now we have access to all this data about fish species, length, coordinates across the Atlantic provinces. So we're able to see like the diversity of fish all over the Atlantic provinces. And yeah, it was just awesome. Like COVID-19, it kind of messed everything up, but in hindsight, I don't think it did, because this was a really great thing or partnership and whole thing that happened out of it. And it also helped the anglers, you know, have some relief in that way too, because they could compete without, you know, being shut down by all the rules. And yeah, I basically just threw in there like, you know, competition for citizen science has proven itself, you know, the best. So challenges influencing our data. So that sounds like issues, but actually they were good challenges. So basically, to avoid any gaps in data, like as the challenge was going on, if we were seeing too many striped bass, which there's never too many, but if we were seeing a lot of that and we're looking for other species, we were able to create challenges for different species, which I mean was good, because then we would be seeing different things come in. And also just random fun challenges like the weirdest catch, or, you know, just like the smallest fish too was really good. And so what I gathered from that was, you know, do challenges throughout the week, do challenges on the weekends, do through full weeks, like mid-weeks, just because, you know, we don't know where everybody is. Some people may be working all the time, or they may not. So it was good to just, you know, have it overall anytime. And there was challenges for diversity. And again, like I said, smallest or specific length. So if we were seeing, you know, a lot of small ones and a lot of huge ones, and we need some in here, then we were able to say, okay, you'll win a prize if you get it in between this length and this length. And that worked out pretty well. And I found that it encouraged anglers to fish out of their comfort zones or within it. And I like to think that it diversifies anglers just by holding challenges that were not striped bass. Because I mean, everybody loves to fish their favorite species of fish, or catch, as Jeff would say. But yeah, you could just go and catch like random things and just kind of make it fun. So overall contribution of the data. So this is a whole bunch of fish that had come in, and like that's just a minuscule amount of the fish that came in. So from May 14th, 2020 to October 31st, 2020, we had 109 participants in the challenge. We had 5,612 fish entries in total, 46 different species, and 38 different species caught by one participant alone, and then 51,895.5 centimeters total length caught by one participant on their own. So like that is how much I know it's like length, but that's how much information is coming in from just one participant. And then we had 2,804 striped bass entries, 1,154 smallmouth bass entries, and 735 chain pickerel entries. Like, that's a lot. So I thought it was a great success. So yeah, I just throw in my acknowledgements, and obviously all the sponsors are there too. I always recognize Sean, obviously, and also his whole team was great the whole time. And Danielle, Rachel, Josh. And then I just put in some hero shots from the anglers that they sent in at the end of the challenge, just to show you, you know, it was fun. So thank you, Bilal, merci. And that's it. So hopefully I didn't go over any crazy time slot.
Thank you, Leah. That was great. Lots of information, and it looks like you had a blast.
I did. So good. I don't know how to get out of this stop sharing. Okay.
You did a good job.
There you go.
Good job, Leah.
Oh, thanks. Okay. Stressful.
All right. Before we jump into the questions, I know there are a couple to do. We got one more poll to do, and somebody, Shannon Brake, asked to explain why we're doing these polls. And so after we do the next poll, I'll give everyone an update on sort of what we're trying to get out of the poll and sort of our goals for that. So I'm just end the last poll and basically share the results. How would you like most to see this tournament data used? Fisheries, well, for management, again, coming out on top. Conservation number 2, and close in fisheries research number 3. So we'll finish off with the final poll now, and then at the end of the Q&A session, I'll give everyone an update on what the results for that is, and sort of give it a little bit more background. And I didn't really have a chance to explain it earlier, so I'll jump in and describe that a little bit more at the end of the talk. So I guess Danielle, you want to sort of stick handle the questions from here?
Yeah, sure. So if anyone has questions, you can put them in the question and answer box. If you're watching this on Facebook, you can put your questions in there and I will get a message letting me know that they've appeared. And so there's a couple questions that came up after Trevor's talk that I'll pass along to him. Leah might have answered one of these already. I'm uncertain. But what is the farthest away a tag has been reported? Do we have any of our tags caught in the US?
Not recently. There are a few tags from earlier studies done in Minas Basin that were fish that got caught down in the US. So we know that for sure. And the opposite, fish that were tagged in the US getting caught up our way. But it seems that the Minas Basin is a real bottleneck there. It's where fish just can't get through. So there's very few, it seems, very few US fish, and the genetic information supports that. US fish that come into Minas Basin. It seems they come up by St. John River a lot, over to Annapolis. It seems that might be historically a place that a lot of US fish went. And it doesn't seem like Canadian fish go down to the US much. Lita and myself are actually looking at historical records of tags with a group in the States called, oh geez, I can't remember what it's called, the Littoral Society. And they've been tagging fish for 50 years or something. And we want to see whether there's any patterns to this, whether maybe population sizes influence where fish go. When populations get big, fish move further, that kind of thing. We saw that when the Miramichi went to, you know, nearly 1 million fish estimated. All of a sudden, there were fish in Labrador. The fish have stayed in Labrador; they're still up there. So this is called range expansion. Climate change is also going to do stuff with that. So we might see more tags in the future being caught from the U.S. But the Canadian fish down in the U.S., we don't seem to get any tag returns. So not recently.
And then there were a couple questions. I'll kind of combine them into one question. But do you have any thoughts about what impact the striped bass and the Miramichi might have on salmon? There were some diet analyses being done. You mentioned that they might feed on Atlantic salmon. Can you kind of elaborate on that?
Yeah, so there's been quite a bit of research done on that. It's not necessarily the area we're concerned with. We're more concerned with developing a way that we can tell what's in a stomach without having to identify it. So you can appreciate that when a fish eats something, and when we eat something, that your food starts getting digested very quickly. And if you don't actually open up that stomach quickly enough, you'll never identify what that fish is. Sometimes you can by looking at the otoliths, because they're different for a lot of fish species. But again, you have to actually find them. And that can be difficult. So we've developed an eDNA method that we call gastric DNA, where we actually get the stomachs, we preserve the goo in the stomach, and we can do this with just a stomach that has absolutely nothing discernible in it, and be able to tell different species that are in there. From that work, we've only found one salmon in our Labrador samples. And I think we have about 29 fish or something like that, 21 fish that we did from Labrador. And there's only one salmon in all of the diets stuff that we've looked at. Well, no, I wouldn't say one salmon, the presence of salmon in one fish, and it was a very small percentage of the diet. So I don't know what the effect is. I know other people are doing a lot of work on the Miramichi and looking at that interaction there. And I think that there's, well, it's right now it's early days, I think, to really know what that impact might be.
Trevor, didn't we have some studies that showed around 2%?
Yeah, again, there's lots of
DFO put out, like, out of 1,480 80 bass stomach contents sampled, it was like 2% or something like that.
Yeah, I should have left I had 3 slides at the end of this talk on all that, because it was a talk I gave at the Blomidon Naturalist Society to answer that specific question using those studies. That's very, very low numbers in the stomach content analysis that were done. Again, though, those are traditional stomach content analysis. That is, open it up and see what's there. So you have to have some discernible body part to do that. So there's some gaps in that, and I know that some other people are looking at sampling more to fill some of those gaps. And there's also with the acoustic information, so there's some acoustic tagging done, and then to look at the patterns in how a fish moves are different in different species. And so you can kind of infer that if you're eaten by something, you start moving like it. You know, if we all of a sudden started moving like a bear, somebody would say, hey, that bear might have a human in it, that kind of thing, if we were tagged. So the same idea. And from that, there's some pretty broad estimates. The problem is the sample sizes are really low. And so, you know, one out of 10, is that 10%, or does that represent something that's very different? It's very
That was the salmon groups put the tags on the smolts, and they figured they got eaten, started heading upstream again. They said that must have been a striper. I said it was a seagull. I'm thinking seagull's heading.
Yeah, there's just a lot. There's just a lot to it, and so it's really direct observation is probably the best way to go. But then you have to kill a lot of striped bass to get the direct observations. So that's where something like partnering up with a group like we're running, where we take the carcasses and can get as much info. You don't need the carcass anymore. You got the fillets, right? We can take it and get lots of information from it. And so that's where we're sort of are putting our cards over the next few years.
All right, we have, I think, 4 more questions that have come in. So one of them, maybe Jeff, this is more for you, I'm not sure, but when is a good time of year to fish for striped bass in the Pettit-Kodiak River?
I'm not sure. I don't know. Trav, you got an idea on that?
Could you just repeat that again?
When's a good time to fish on the Pettit-Kodiak for stripers?
It seems that they come up there later in the summer, around in August. That's the data over the last 5 or 6 years shows that the larger fish come up there in August. There's some smaller ones that are around earlier than that, but fishing wise.
Great. And then Victoria is wondering if we consider climate change as something that's anthropogenic, so, you know, caused by people, then why wouldn't we call the range expansion for striped bass an invasive species?
Oh, we would. That's so invasive species is a term that has there's a lot of nuances to it. That you have invasive species and you have alien invasive species. That might be the simplest split. Alien invasive species are ones that shouldn't be in that area. Invasive species sometimes just means when you have an explosion of a species in an area, and so it becomes invasive from the sense of there's just too many of them here, and naturally there wouldn't be that many. So yeah, that's precisely what's happening in Labrador. Is this range expansion is initially as an invasive species. There's no regulations for striped bass in Labrador, so people initially didn't know what to do with them, so I suggested they barbecue them because they're tasty. And so that's what they're doing now. So we're about 3 or 4 years on from that, and but there's still no regulations up there about what to do with striped bass. So those management rules are lagging here in this case.
Okay, there's one more in the Q&A box and then one in the chat. So if you catch a striped bass and you're planning on releasing it and it's a large specimen, if you hold it vertically versus horizontally, does it change its capability or its likelihood of surviving?
Yeah, probably. There's a lot of catch and release information on other species, and it's usually done at tournaments or derbies, looking at the stress levels in fish, looking at mortality levels, impacts on the fish, those sorts of things. A large fish, because it's in water, is buoyant, and if you turn it upright, all of that heavy mass of the internal organs, which are not well supported in the body. Our internal organs are well supported by lots of different connective tissues, and so our heart stays where it's supposed to be, etc. In a fish, it's less so. And you can ask Leah or some of the other students that have dissected a lot of these fish. I mean, there's not a lot besides the actual organs that are sitting in there that's actually holding them in place. So when you move them around, those organs are going to compress on each other, and that could cause issues. So it's best to support them as well as you can under both sides.
All right, and then the last question I see is in the chat. So if we are tagging striped bass here in Atlantic Canada and we're not seeing the tags anywhere else down in the States, what is the migration pattern, or what can we learn about the migration pattern?
Well, it seems to be fairly localized. There are a couple of nuances to that, and this is something we're learning just recently in the last 3 or 4 years with more of these acoustic studies, that there are different segments of the population, if you want to think of it that way. So most fish from the Shubenacadie, for example, come into Minas Basin. Some stay in Shubenacadie, some never go into the Shubenacadie. They just stay out in the basin for more of their life, maybe except for the spawning time. There's some fish in the St. John River that come over to the Shubenacadie, spend a month there every year, and go directly back to the St. John River and stay there for the rest of the year. They're Shubenacadie genetic fish, but they spend all their time in St. John River. So there's these little nuances about the populations that are segments of the population that are doing different things. So what's the overall migration pattern? I'd say it's like most anadromous fishes. They come out of the freshwater areas where they're spawning. They spend time in the saltwater where presumably there's more food resources, and they go back up into the freshwater to spawn. Striped bass have this other weird thing they do where a portion of the population overwinters in freshwater areas like deep pools and lakes and things. But some stay out in the saltwater for the rest of the year. It's basically a strategy. So the population has a strategy that if you do different things and something changes in one area, that you still have some of that population is going to survive. Yeah, I hope that answers it. But yeah, there's not a long migration for striped bass in Canada, it seems. It seems the US population moves up the eastern seaboard following all of its bait fish, but the US or the Canada population comes down from the rivers or stays in the bays and waits for those the migrating fish to come in to feed on, like the alewife and the smelt and things like that.
You had mentioned the St. John River, and so there's another question. Is there any confirmation of spawning in the St. John River or of the St. John River striped bass genetically?
Yeah, there's definitely confirmation genetically that there is a distinct genetic population in the St. John River. The part of the problem is we don't know whether that's a remnant of the historical population because there aren't any samples of genetic samples that go back far enough before the St. John River during periods when it didn't have many striped bass in it. There seems that seems to be a population though. The simplest explanation for that it is the St. John River population. Now there's a lot of mixing that goes on in that population though. There's US fish that comes up. There's some interbreeding going on. Nobody has nailed down where the breeding, the spawning sites might be in the St. John River. That's ongoing work right now. There have been small fish found there though. And those fish actually come out to be that genetic type of St. John River. So, you know, all the indicators are that something's happening there. It's just nobody's nailed down where it's happening.
Great. All right. Well, that I think that's the end of the questions from what I can see. So I'll pass it back to Sean.
Well, thanks, Trevor, and thanks, Danielle. And great questions. We're wrapping up the final poll here. I'll just end it and we'll share the results. And so we want to share more data with you. What type of data would you like to see on the app? Number one is fish movement and tagging, followed by catch rate, which actually is already on the app, but we don't widely distribute it. If you report your catch out, you'll see your catch rate on the app and then what the average is for that water body. But we can certainly look at doing a lot more of that. Catch and release data. And then tied for last place is size, range of fish, and the species of fish or a species of invasive species. That was awkward wording there, but anyways, I appreciate everyone filling that out. And I think it was Shannon's question, sort of why are we doing these polls? So I'll step back a little bit and tell you a little bit about how these tournament talks have started. And they really kicked off last year at the American Fisheries Society conference. I was holding a symposium on angler reporting and data validation. And we realized that where we're not doing a good job is reconnecting with the anglers once the data is collected. And if we're going to count on having anglers continue to report their data in the future, we need to do a much better job in making that connection back with the anglers and developing meaningful connections. So that was sort of the foundation for these tournament talks. And so being a scientific organization, we want to think about this from a scientific perspective. So we treat these like experiments. So what can we learn from anglers during these talks? And so these polls are a way for us to standardize and get measurable information from anglers during these talks. And that's really the objective. And also to find out what do we need to do in terms of the data that we're generating, being able to share that back with the anglers. So long as we're not revealing secret spots, that is a must. No go, can't go there, third rail, big mistake. But there's a lot of other data we can share back, such as the catch rates. If we're able to tie in tagging information, let's say from Trevor's project, and there's a number of other fisheries researchers we work with that have tagged fish, maybe there's ways we can tie some of that data back. So anyone that reports a tagged fish can then see all the tagging information related to that fish. And we know that is something that's really valuable for the anglers as well. So this really is a good education for us to figure out, based on the questions that are being asked, based on the poll results, that are coming up. And we will also do a follow-up survey just to get a sense of what you think about these tournament talks. How can we improve them? How can we help deliver information back to you in a more coherent and logical way, so you can quickly access it rather than digging through reams and reams of data reports. So that's sort of hopefully, Shannon, I answered your question. If you have any other questions, feel free to make a post, and certainly you can reach out to me. I'll put my email address in the chat for anyone that wants to reach out to me directly and talk some more. And I guess I'll put just maybe some final words to Jeff, who was the angler that we connected with on this. So Jeff, off to you.
Well, I didn't know I was going to be back on the agenda, but no, that's great. Well, I'd like to thank everybody, Leah, for your work behind the scenes and your whole team. It's been really great. And Trevor, you being a leader down there, and Sean, this is, I think it's been a great summer of learning. And we certainly, from my perspective as an angler, not a scientist, these things just is an incredible amount of information for me. And but most anglers don't care about gonads. We learned about that today, that they're not that at the polls. We're not care about gonads. We don't care about the fish. Just tell us what you can. But it was a great, and we certainly, I certainly hope to think that we can do this again next year. That we get enough participation and we can find volunteers. Leah, I don't know what you're doing next summer, but hopefully you're prepared right on. But I'd like to thank. I like to thank everybody for their help and all the participants. But we had 100 and how many?
109. Sorry.
109. So we didn't really know what we would get, to be honest with you. But I think at the end with the leaderboard, and that it was getting a lot better. So I'm good. I thank everybody for tonight. Trevor, you want to say any last words? You certainly were the star of the show tonight. You and Leah, the science was everything. It always is.
Yeah, it's always about stripers. Anybody can contact me anytime. I'm quite available. stripebass@acadiau.ca is one of my email addresses. Our Facebook page will lead you there, Striped Bass Research Team. And I monitor quite a few of the fishing groups and I participate on those. So I'm quite available. Anybody wants to call, let's go for it.
Okay. Sean, I think we're good. We went an hour and a half instead of an hour.
No, well, we were planning an hour and a half, or at least I was. We discovered
were you? Okay,
the last
perfect.
time around. Yeah, an hour was too short because when people want to start asking questions, we really have to make sure we can answer them in a time.
Yeah, well, that's good. So we're right bang on. Perfect.
Spot on. Yeah, everyone, great job. All of Trevor, Leah, and Jeff, really fun to work with you, and looking forward to a great.
And Daniel, of course, in the background. She's not saying much back there. Keep sure.
All right, everyone. Real pleasure. Tight lines, and we'll pick it up again shortly. Take care.
Okay. See you. Yeah.
Thank you.